This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 270, recorded on July 21st, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And from Ann Arbor, Michigan, Michelle Swanson.
Hi, it's great to be back with you.
And we are in the heart of summer, right?
We are indeed.
We are. I don't know how it got to be the end of July.
And it goes fast.
My goodness.
I mean, you get to the 4th of July and you blink and it's August.
Yeah, pretty soon we'll be back in September. Michelle will be going to football games.
Go Blue.
But we are always here doing microbiology for you so that you can, any time of year, learn about these amazing microbes. And today we're going to do the same, and we're going to start with a snippet from Michael.
This is a bit unusual. The title of the paper is Coupling Magnetic Field and Salinity Upshock to Improve Polyhydroxyalkanate Productivity by Halophilic Mediterraneae, Feeding on Molasses Wastewater.
What a mouthful, Michael.
It is indeed.
Who knew there was molasses wastewater, first of all?
and more importantly, that magnetic fields can actually influence fermentation yield. And it's a paper that appeared in Applied in Environmental Microbiology in June of 2022, and it's by Chen, Wei, and Wan. And this manuscript is really pretty significant because it concerns enhancing the production of a green plastic precursor, the polyhydroxyalkanate, or PHA, and that's what I'm just going to call it from now on, PHA, by enhancing a fermentation yield from, of all things, an Archaea, Halophilic Mediterraneae, through the manipulation of the saline concentration that the microbe finds itself in, and by manipulating the magnetic field around the fermentation vessel during the fermentation process. And first off, PHAs are synthesized from the available intracellular pool of carbon and energy sources during growth limiting conditions. And they accumulate secondary to stressors that are placed on the bacterium. Now, PHAs are a well-known family of bacterial-based biodegradable plastic precursors and offer an approach to carbon neutrality and support a more sustainable plastics industry. It's because of their biocompatibility and their thermoplasticity that they are this attractive alternative to petrochemical-based plastic precursors for the creation of plastics. And we've already talked about the 5 gyres that are floating out there in the ocean, the plastic islands that are growing off of our shores. And it really puts it into perspective why we need to do this. So why aren't we using PHAs? And the answer is simple. It's economics. It's because of the high production costs that these green precursors are still limited and their ability to be produced at an industrial scale.
And Michael, if I could interject, it is no coincidence then that this work is coming from a group at the National Engineering Laboratory for Advanced Municipal Wastewater Treatment and Reuse Technology in Beijing at the University of Technology in Beijing.
And I'm sorry, I apologize for not pointing that out earlier, but thank you, Michelle.
Yeah,
because
applied research.
That's where we get the molasses waste. So enter this group and their quest to develop an approach to produce a low-cost PHA pipeline for the green plastics market. And the microbial contribution to the green plastic pipeline involves this enhanced production of PHAs through fermentation by, of all things, halophilic microbes. Now, if you take a step back and ask yourself the first question of why halophiles, and the answer is that the fermentation of PHAs can be accomplished then under non-sterile conditions, because these suckers grow in 200 grams per liter sodium chloride.
Yeah,
that's
which is where the term
salt.
halo refers to their high salt tolerance, right?
So you don't need a sterile fermenter. You don't need sterile feedstocks, and the recovery can be achieved by osmotic lysis of the cell. By using these non-sterile conditions and recovery simply by popping the cells through osmotic shock, the net effect has reducing the cost by more than 20%. So for green to compete, it has to be faster, cheaper, and better. But like all things related to biotech, and I think Daniel said this in one of your TWiM clinical updates, you can only generally have two of these: fast, cheap, or better. And here they want all three. So what's the other issue in producing green plastic precursors? That right, we need to consider the carbon and energy source that goes into making the precursor. This normally accounts for between 30 to 40% of the PHA production costs. But if you simply repurpose a waste stream into carbon and energy, and in fact, sometimes folks will pay you to take their garbage away for them. And this is the case here in the manuscript that we're going to discuss today, where they're actually using a waste stream. Now, the experiments that the authors set up for us is they're simply looking at production of the microbe and production of PHA and how much PHA is actually produced. And that's effectively volumetric productivity. It's generally the third panel in all of their figures, because the higher the volume per unit means you're getting increased production efficiency, and it also goes to reducing the size of the equipment you need in order to make the precursors. So now you have the goal of these authors' paper. How can you increase fermentation productivity while at the same time keeping costs low? So let's consider the microbe, Halophilic mediterraneae. It's the PHA producer from the family Halobacteriaceae, and this particular microbe can accumulate a whopping 65% of its dry weight using glucose as the carbon source. But who wants to pay for a carbon source? We want to use something else, enter the wastewater. But the trade-off here is wastewater fermentation has low PHA volumetric productivity. And I dropped a nice article into the show notes about this remarkable archaeon, Halophilic, so you can take a look and read up on the biology of this microbe. The studies that the author present focus on the optimization of the fermentation yields via the optimization first of growth conditions such as temp, carbon and nitrogen ratios, and nitrogen sources, and a bit of the biology about PHA. It accumulates within the cell under stress conditions that limit the growth of the microbe. But it's a double-edged sword. The conditions favoring PHA accumulation also reduce biomass proliferation, and hence the double edge, because you need biomass in order to accumulate massive amounts of PHA, leading to a low yield of biomass under the stress conditions. So they wanted to enhance this volumetric productivity. That's their goal. Enhance volumetric productivity by effectively stressing the microbe. And the stressor that they're going to use is, of course, salt. Who would have ever guessed a halophile would be afraid of salt? And instead of using 200 grams per liter of salt, they up that concentration to 300 grams or 30% salt.
Wow.
And they also stumbled onto a paper by Fetedi and Ati that observed that when a low-intensity magnetic field was placed around microbes, this could stimulate the enzymatic reactions by altering the electronic spin state of enzymes. And if you think about basic microbial physiology that you learned once upon a time, you know of the need for transition metals. And basically what they facilitate is redox shuffling. They take the high energy hit from the electron, enable it to calm itself down a bit, and then transfer it in the electron transport chains. And that's what Fetedi and Atai showed, is that a 7 millitesla magnetic field. And for those of you that have forgotten your college second semester physics,
or never took
a magnetic
it in the
field
first place.
is nothing more than a vector that describes how a magnetic influence can move electric charges and electric currents. And one central theme to recall from metabolism, it's all about moving electrons and the efficiency of moving electrons. And let's face it, bacteria are continuously moving electrons about. And I went to Rich Condit's favorite source, Wikipedia, to introduce you to magnetic fields. And I dropped that into the show notes. But to put it into perspective, there was a paper by Stansell that described a 60 millitesla field magnetic field enhanced resistance in E. coli upon exposure to piperacillin, an antibiotic. So you might be wondering what this unit of measurement, a millitesla, means. And for the sake of time, I'm just going to tell you a millitesla the millitesla concentration that they're using in this paper is about 1/30th to 1/60th of the strength of your garden variety medical grade MRI. That are principally two flavors of MRIs in your modern hospital, a 1.5 Tesla MRI and a 3 Tesla MRI. And having just done my annual training, I know you'd never go near a MRI because it'll literally rip the metals out of you. And so it literally is having an influence on any enzyme in the bacterium that requires metallic cofactors. And again, a rule of thumb is anytime you hear the word hydrogenase, you can bet your ranch that there's a metal involved in the transfers of electrons. So the authors postulate that based on studies like using magnetic fields, they could potentially increase biomass productivity and resistance to adverse conditions. Now you know I was curious about this paper. And in fact, the authors offered, no one has shown a magnetic field effect on the Archaea. So their candidate microbe is an Archaea. So this is going to be novel. And they're attempting to optimize PHA to help supply the green plastics industry. So the data they present are relatively straightforward, easy to digest, and they walk us through their exploration of feasibility of coupling a magnetic field with a hypertonic condition to enhance the PHA volumetric productivity. So the first question they asked is, does increasing the salt concentration from optimal of 200 grams to 300 grams increase the PHA concentration? And the answer is yes. I mean, it's a really easy graph to look at. And more importantly, you look at it, you understand the volumetric. And in fact, metabolomic studies by others have shown that under conditions of non-optimal salinity in these halophiles is you have reduced cell growth and reduced cell division. because of downregulation of metabolites related to glycolysis due to the stressors, the TCA cycle, branched-chain amino acids, and of course, DNA replication. But the second panel of their first figure offered that the product that they're interested in, the PHA, was highest when the cell was growing in 300 grams per liter of salt, where 62% of the intracellular material was PHA. So that's a lot of PHA. So the PHA cell content increased in these elevated or non-optimal growth conditions. So now you might be asking, so how can we optimize growth? And what's actually going on? Well, there's an old observation about living in high salt, and it's called upshock saline environmental adaptation. And the way the halobacteria do it is by producing two proteins, beta-keto-acyl carrier protein reductase and 3-hydroxyacyl coenzyme A dehydrogenase, which, as you might surmise, resulted in enhanced synthesis of PHA. Now, the puzzling thing to me was their third panel, panel C. So how do you get the enhanced productivity? So here's where they toss in the magnetic fields. So in the saline environment, and they use a variety of magnetic fields from 10 millitesla all the way up to 70, and just cutting to the chase, the 50 millitesla field is the sweet spot. That's where, when you increase the salt concentration to 300 grams per liter and you have a 50 millitesla field, you can increase the concentration of PHA. Now, the next question on your mind is, how is this all working? And in accordance with previous research showing that salinity increased PHA accumulation, they asked fundamental mechanistic questions of how this might work. How might the magnetic field that they're applying ameliorate the stress of the bacterium upon finding itself in the high salt concentration? And first, as background, the extreme halophilic Archaea regulate their osmotic pressure by this salting in strategy, where they hyperaccumulate potassium ions and chloride ions. And the reason they do this is to retain the functional conformation of the proteins inside the cell at these high external salt concentrations. And this is a massive adaptation that takes place in order to help them live in high salt. And again, the authors show us that when they're growing in this high salt environment under the influence of 50 millitesla magnetic fields, that the intracellular potassium concentration is the highest. So that means it's protecting the normal functions of the cell best. And so these the results then offered that the magnetic field was promoting the accumulation of intracellular potassium to alleviate the stress placed on the cell by the increased salinity.
To balance the osmotic pressure.
They next asked the question, what's actually going on with the cell biochemically? And they found that some intracellular organic substances were up, such as betaine, which is a precursor in this PHA. And they also looked for other things like proline, glutamic acid, aspartic acid, and trehalose. But the betaine was the highest of all of those molecules. And in fact, betaine synthesis was up 23% in the presence of the magnetic field at this 300 gram salt concentration. And that was 74% over what was seen in, if you will, the wild type or 200 gram salt concentration absent the magnet. And the authors say that it's tempting to speculate that the magnetic field could encourage osmotic adaptation in this halophile to salinity shock by promoting betaine production. And here's
Michael?
where your understanding of how stuff made comes in handy. Most halophiles synthesize betaine by choline oxidation. And choline is converted by, again, aldehyde or choline dehydrogenase and a betaine aldehyde dehydrogenase. And anytime I think of hydrogenases or dehydrogenases, I think of metals facilitating the electrical movement of those electrons. And as the key enzyme, this betaine aldehyde dehydrogenase was explicitly detected in this study. There are other things that are upregulated by the magnetic field in high salt. They actually upregulate superoxide dismutase, which enables it to deal with the oxidative insults from higher metabolic loads. And the final question they asked is, does the magnetic field enhance the production of PHA when they're using this molasses wastewater? And as you might guess, the answer is yes. Salt and magnetic field strength resulted in almost a 50% increase in the maximum intracellular content of PHA.
Wow, that was a good day.
And so what these authors have done for us is they have shown us the value proposition of really outside of the box thinking, of invoking a magnetic field to enhance something that we all sort of learned about as young microbiologists, of the importance of cofactors. We know about adding trace elements to define media, but did we ever imagine that we could manipulate the magnetic field to enhance the ability of a microbe to withstand growth limiting conditions and still make a useful product for us? So this was a really cool paper that I thought the audience would enjoy and get a kick out of.
Hey, Michael, could you put in context for me the 50 milliteslas of magnetic field?
Okay, so the garden variety medical MRI, there's two flavors that are commonly used in hospitals: a 1.5 Tesla field strength,
Okay.
and a 3.0 field strength. The higher the magnetic field, the better the resolution, because it activates more of our water molecules to effectively give it the higher degree of resolution.
But
Recently,
what I'm curious about is, this is 1/20th or
1/30th to 1/60th of the field strength
that would be used clinically. Mm-hmm.
that is used clinically. And in fact, in research labs are often using 7 Tesla magnets to effectively do their NMR studies. Some are even using 9 Tesla magnets to resolve their structures in NMR. Remember,
so
NMR
people are being pulled by their belt buckle into this magnet when they're trying to do these experiments then?
No. It's and as I said, I had just completed my annual training, because we all have to be aware of where the MRIs are in our facility, and we know not to enter a magnetic facility because we could be pulled by our belt buckles. And any of you who ever have done hobbyist welding, you sometimes have metal fragments in your skin, and they can literally be pulled out of your skin. And the welders are most concerned about the metal fragments. And these are microscopic metal fragments that they have in their eye that can get pulled through their eye and literally.
Yeah.
Oh my gosh.
Or a nose piercing. That would be uncomfortable.
Oh, nose piercings.
So Michael, the other thing Vincent and I were wondering about is this molasses wastewater. And I've just looked on the World Wide Web, and it says it's a high-strength effluent of the food industry, where distillation is involved, and it can also occur during sugar and yeast production plants.
And so and it adds to the biological oxygen demand of your sewage treatment plant. And I know one state in the United States, California, actually charges producers of waste streams that are high in BOD a higher tipping fee in order to literally flush it down the drain because of the impact it has on the BOD of their
Sorry, what's BOD?
biological oxygen demand. The higher the carbon load, the bigger the surface area the plant will need in order to biologically degrade waste so it doesn't cause algal blooms in rivers and all of those other things.
So there are really wonderful implications, practical implications of their discovery here.
So and you have making plastics and also getting rid of this molasses wastewater. Yeah.
Mm-hmm.
I mean, this is a really interesting technique because also you're using a halophile where you don't have to worry about sterility. If this was in, a garden variety bacterium that we routinely use in the lab, everything would have to be sterile. You would have to use aseptic handling techniques. Here, because you're using 30 grams per liter, 300 grams a liter of salt.
Yeah.
I mean, you're really pretty in good company. Nothing much is going to grow in 300 grams per liter of salt.
That's great. That's a good point. This is a multifaceted advantage if they can get it to work. I hope they
have to scale it and, but there's enough ancillary literature out there encouraging magnetic fields and looking at this. So this is a brave new world of microbial physiology.
Nice.
Very cool. Thank you.
Good. Thank you, Michael. That was great. I really, I learned a great deal there. All right. Now I will talk about a paper actually Michael found, but he couldn't get a hold of the preprint, so I decided to take over. And it's published in Nature Microbiology. Bacteria deplete deoxynucleotides to defend against bacteriophage infection. First author, Nitsan Tal. The last author is Rotem Sorik. And I was in the Madison airport yesterday, and I emailed Rotem, and within minutes, he's in Israel. He answered, yeah, I'd love to. Here's the PDF, and I'm honored to have it discussed on TWiM.
And Nitsan also replied to my request for some biographical material, so I'll be weaving that in.
And I'm so happy we got it because it is so cool.
Well, in the meantime, Michael decided to do the paper you heard, which is very interesting. And now you'll hear this one, which is also extremely interesting. So everybody wins. So this is all about bacterial defenses against bacteriophage infection. And we have actually talked about this before, most recently on TWiM 265, where we talked about hotspots of anti-phage systems in bacteria. And this was how these authors start that defenses. There are a lot of defenses in bacteria against phage, as you might imagine, because phages are ubiquitous and they can kill the bacterium. So there's been an arms race, an evolutionary arms race on both sides. Bacteria develop defense systems, phages try to overcome them. Restriction, modification, CRISPR-Cas systems, and many others. And a hallmark of many of these is that the genes encoding the bacterial defense systems tend to cluster on the bacterial chromosome. And in the paper we did on 265, they actually identified new defense systems by simply saying, what gene is next to this one that we know is a defense system?
And this group, Nitsan Tal and Rotem Sorik, recently published a snapshot in Cell that has a really great graphic that describes the multiple defenses that have evolved in bacteria to protect against phage. So I've included that in the show notes so that for those of you teaching or curious about it, it's amazing what this arms race has generated over the eons.
So they start by looking at a family of genes in many bacteria with a predicted cytidine deaminase domain. All right. Now, cytidine, of course, is one of the 4 bases that make up DNA. And if you take a amino group off of it, it becomes a different base. And so that's what it's a cytidine deaminase is simply the enzyme that will remove the amine group from the C and make it into a U. And they said they looked at these genes encoding these cytidine deaminases because they're next to other phage defense systems in bacteria, which, as I said, is, as they say, a strong predictor for a role in anti-phage defense. So they take these genes, they take 2 of them, one from E. coli, 2 different strains of E. coli, and they put them into a lab strain, the famous MG1655, which both of you, I'm sure,
yes,
use all the time. I don't use E. coli anymore. And then they ask if you infect MG1655 with this cytidine deaminase gene, what does it do against phage infection? And in fact, it confers defense against a diverse set of phages. Let's tell you which phages here that they used. Figure 1B. It's a whole panel like T2, T4, T6, T7, T5. All right. So it seems to confer defense. And if they change the active site, so there's a deaminase part of the protein, it's the active site. They can change a single amino acid so it's no longer active, and it abolishes defense. So it sounds like the deaminase part of this protein is important. There's also a nucleotide binding part of this protein because, of course, a cytidine deaminase to work has to bind a nucleotide. And so if they change the nucleotide binding motif, it doesn't work either. So this looks like it's a defense protein, a cytidine or cytosine deaminase. And these have been found in other systems, and particularly in human cells. We know that there are cytidine deaminases that are antiviral. For example, the ApoBec. ApoBec is the name for a series of genes that encode proteins that are cytidine deaminases. And some of them are directed against HIV-1. They change C to U, and therefore you get a lot of mutations in the viral genome, and the virus is no longer infectious. All right, so they extract total DNA and RNA from deaminase and control strains of this E. coli. So remember, they put the deaminase genes into it. So they have those and control strains without deaminase. And they don't see this C to U or C to T as you would see, say, in an ApoBec, which changes C to U and mutates it. This one doesn't seem to do that. So it must be a mechanism that's different. It's not a deaminase in itself. It doesn't involve hyper-editing, which they call it, mutating the genomes to death so they're no longer infectious. But there's some other activity. So the ApoBecs do that. And in fact, they have been called weapons of mass deamination. WMDs, right?
These are, and it is the bacterial equivalent of a WMD.
It is, but it's not a WMD, right? Because they don't find an increase in C to T in a E. coli producing this cytidine deaminase. So there's some other mechanism. So what is it? So that's what they're going to look at now. Next, they ask whether the enzyme modifies single nucleotides or does it modify polymers of DNA and RNA. So they take extracts of cells that are infected with T7 and they use mass spectrometry to look at the nucleotide content. And they say, remarkably, whenever you see remarkably, you got to get ready, right? Remarkably, DCTP. So when phage T7 infects the cell, phages need a ton of triphosphates, right, to make more genomes. So the synthesis of triphosphates goes up. DCTP was completely absent in infected cells with this deaminase gene.
Completely absent.
I bet it's not completely. It's got to be something there, right?
The data are so clean. Wow.
Yeah. And as you take away DCTP, what goes up? DUMP.
Yep.
Now you're asking, wait a minute, why doesn't DUTP go up? Well, because there's an enzyme in E. coli that converts DUTP to DUMP.
And it's happening very quickly, within 5 or 10 minutes. Boom.
Oh, yeah.
Converting.
These phages are quick.
Well, T4, T7, has a burst, an eclipse period of 40 minutes. So everything has to be soup to nuts in 40 minutes. So it's the entire menu in 40 minutes. They inject and they're done.
Yeah, that's right.
But in this case, it's the bacterium that is so quick.
Oh, yeah.
It's the bacterial response that is within 5 or 10 minutes. Boom. They've converted all the DCTP.
And so this is specific for CTP. So you don't see CTP, CDP, CMP changes. It's deoxycytidines, DCTP. All right. So the idea here is that if HT7 infects, there's a defense protein, this deaminase. It converts DCTP into DU. It depletes the cell of DCTP, and therefore the phage cannot make DNA, right? And so it's impaired. The other three triphosphates, the DA, GG, and DT, they all accumulate normally in the cell. So this is specific for DCTP. And the reason why the others actually go up, the ATP, DGTP, and DTTP go up, and the reason is, well, when T7 infects, it turns on the synthesis of all 4 triphosphates, but it can't replicate DNA because there's no DCTP, so there's an excess of the 3 other bases. Makes perfect sense, right? So they would have normally been incorporated, but they're not. So cytidine deaminase is a cool defense mechanism. It gets rid of DCTP. The bacterium is going to die anyway, right? So why not take away DCTP and stop the phage? It's kind of an altruistic
That's right.
effect, right?
Yeah. Protect the population.
Now, are there other mechanisms for depleting DTP? And now in eukaryotic cells, there's a protein called SAMHD1, which restricts HIV replication by removing the triphosphate from DNTPs. All right, so you have phosphate-free deoxynucleotides, and then you have inorganic triphosphate after SAMHD1 works on them. And this is a massive degradation of DNTPs. And the virus cannot replicate because you need DNTPs in order to make genomes. So are there such things in bacteria? And they say, yeah, people have found DNTPases in bacteria, but then nobody has worked on them all that much. And they say, well, maybe they are phage defense proteins. So they identify these bacterial DGTPases. All right. So these are enzymes that would remove the phosphates from GTP. They found 8 genes. They took all of them. They put them into E. coli MG1655, and 6 of these 8 clone genes protect E. coli from phage infection. And a change in the active site abolishes the defense. The active site is the GTPase site, and saying that it is important, in fact, to remove the triphosphates. And interestingly, if you overproduce the GTPase, it doesn't impair bacterial growth. So they say the defense is not simply an artifact of overproduction. So they then go back to mass spectrometry to look at nucleotides in cells that are producing this DGTPase. And again, when you infect cells with T7, 10 minutes later, you get elevation of DGTP levels. But if you have the DGTPase, boy, that's a hard one to say 10 times fast. DGTPase. DGTP is present in lower concentrations throughout infection. And you see elevated concentrations of deoxyguanosine, right, which is without the 3 phosphates. So guanosine triphosphate, or guanosine is the base and the triphosphates on it. So you are taking the triphosphate off of the GTP, and so you see elevated amounts of deoxyguanosine. And again, all 3 other DNTPs accumulate because phage DNA replication is impaired when you take the triphosphate off the DGTP, so can't replicate DNA. So all those 3 other bases, all the triphosphates can't be used, and so they accumulate. So now they find a second family of antiviral genes that deplete DGTP.
And again, it's incredibly rapid.
Very rapid.
Yeah, 10 minutes after, you don't see any phage DNA.
Yeah. And this is induced
Down to baseline.
apparently by infection. So there's some way, which we'll get into in a moment, that this is turned on. So they look in sequenced genomes of various bacteria, and they can find DCTP deaminases in many genomes, 2.5% of the 38,000 genomes that they analyzed, a variety of different kinds of bacteria. They also find the DGTPase genes in the bacterial genomes as well. So these seem to be present in many different kinds of species, maybe a almost universal kind of defense. Now, if you have a protein in a host that is antiviral, one of the things you always do to get more insight is to select for viruses that can overcome it. And all you need to do is propagate your viruses in the presence of the gene product and see if some can arise that are resistant. So they did
the power of classical genetics.
Classical genetics.
Yeah.
They obtained 4 mutants of T7 that could partially overcome defense by DCTP deaminase and 7 T7 mutants that overcome DGTPase defense. So again, these are mutations in the phage genome that make them less susceptible to these bacterial defense proteins. So where are they located? For the DCTP deaminase, they found changes in gene 5.7, and I'm going to tell you what 5.7 is in a moment. In the old days, we used to name genes. We were very straightforward, gene 1, gene 2, gene 3. And in particular, we didn't know what they did, so we
right.
just
used numbers.
It's much different. And then 3 of the mutants that escape the DGTPase were also changed in the same gene, which is really cool. They also find some changes in a gene called 5.5, and one T7 was not changed in either, but had a change in an endonuclease gene, which is important during DNA synthesis. And so this 5.7 gene seemed to be allowing escape of both the DCTP deaminase and the DGTPase defense. This escape is not full; it's partial escape. So these changes do not fully overcome the defense. But they do so enough to be able to reproduce. Now, what are these genes doing? They look at 5.7. Now, gene 5.7 in T7 encodes a protein that shuts down a particular type of transcription in the host called sigma-dependent host RNA polymerase transcription. So sigma is a transcription protein or a transcription factor that's needed for the activity of this DNA-dependent RNA polymerase, which makes the messenger RNAs that are eventually translated to produce protein.
It gives promoter specificity.
They're thinking that somehow transcription is involved in turning on these defense systems, right? Because if you're not infected, you don't need to turn on deaminases and GTPases because that could be detrimental in itself. You only want to do that if there's a virus around. So they think and in fact, when T7 infects the cell, it inhibits transcription of the host cell so that it can have access to all the transcriptional machinery to itself. So they're thinking maybe there's a connection between transcriptional inhibition and the induction of these defense systems. So they with that hypothesis, they do an experiment and they use a drug called rifampicin, which inhibits these DNA-dependent RNA polymerases in bacteria. And they use uninfected cells with or without the deaminase. So deaminase, the CTP deaminase or the GTPase. So rifampicin with these, and the nucleotides become depleted in the presence of rifampicin. There's no phage here. All they're doing is inhibiting DNA-dependent RNA synthesis, transcription. with
I
this
would have
drug.
felt brilliant the day I did that experiment, saw that
Yes,
result.
it's a good experiment.
It's so clean.
I mean, it's night and
And in
day.
fact, it turns on this bacterial defense system. So inhibiting transcription seems to be the key. A phage inhibits transcription. This drug inhibits transcription. So that both of those activities seem to turn on the production of these two proteins, the deaminase and the GTPase, that inhibit phage. So, how that actually works, they need to sort out, but that's an interesting clue. That would be a signal to the host cell, right? Because what the phage does is inhibit transcription. So the cell can sense that somehow and turn on these systems.
So the phage can take over the whole transcription components, right?
Everything, the triphosphates, whatever other components it needs. Yeah.
Shift it from the host to the phage.
The phage, yeah.
But then the host says, not so fast.
Not so fast.
We're going to deplete your building blocks.
Right. So that's the story. Infection by phage triggers activity of these nucleotide manipulating enzymes. They deplete just one of the four. That's all you need. And now the phage can't reproduce, and that ends the infection. And they think that inhibition of host transcription is the trigger for this. So I'm sure they're working on it, and they'll tell us more. And I just want to point out before I stop here that enzymes with cytidine deaminase are known to be involved in several aspects of immunity. So we have the apobex that protects cells from viruses by deaminating cytosines. We know that there is a cytidine deaminase involved in somatic hypermutation that is responsible for antibody diversification. So when you get a booster vaccination, you're making various different kinds of antibodies as a consequence of a cytidine deaminase that changes the sequence of the antibody gene. So you get slightly different proteins, and sometimes they're even better. And now here they say this is a third role for these deaminases, where in bacteria they can deplete the triphosphate pools so that the viruses can no longer infect. So really very, very exciting. And
really exciting. And I was so impressed when they did their survey to ask how prevalent this class of enzymes are. And they found it, as you mentioned, in lots of different bacteria. But within any one genus, a small percent of the strains actually had it, which they could then deduce that these are probably moving by horizontal gene transfer and being shared in the microbial world.
Right.
So this is really widespread and so beautiful.
They conclude by saying, we find it remarkable that depletion of the nucleotide pool is a successful antiviral defense strategy shared by eukaryotes and prokaryotes alike. And, the mechanisms are similar. There are differences. It makes you wonder whether the common ancestor of eukaryotes and bacteria had some related system because there are already viruses infecting them at that time, right?
That's certainly the simplest model. Yeah. Such beautiful work, and done so cleanly with their directed site-directed mutagenesis and then their classical mutagenesis. Let's look for escapee mutants. And in each case, they hit pay dirt. It was really great. So it's a large group, but the first author is Nitzan Tal. She earned her Bachelor's of Science in the Life Sciences at the Hebrew University, and then went directly into a master's program at the Weizmann Institute, where she started working with Rotem Sorik, and then continued on, and she is currently a PhD candidate there. She's had a really terrific career, has been recognized for a lot of her great work. She, in 22, got a Memorial Prize. In 21, she got a Best Short Talk Prize. She got in 2019, a prize for outstanding students from the Weizmann. She has presented her work at a conference in Heidelberg, Germany in 2021, and also another one at the Innovative Genomics Institute at UC Berkeley, an invited talk. And she's published not only this beautiful paper, but in 2021, they had a paper published in Cell where they discovered a family of pyrimidine cyclase enzymes that specifically synthesize cyclic CMP and cyclic UMP after phage infection, which then activate an antiviral response. So she's doing amazing work, and I am delighted to say she also has found time to volunteer. So she works with an organization that promotes public health by making reliable medical information accessible to the community, especially the parents of kindergartners and teachers of kindergartners. And she also tutors minority students that have academic challenges at the Hebrew University. And now she is enjoying a maternity leave. She and her husband have welcomed their first son, Yali, and she's home on maternity leave, but found time to respond to my email, which I really appreciated. She comments that naturally she's now learning there's a need to balance her passion for research with her devotion to her family. But what has really made this possible is the support and understanding of her partner, but also her mentor, Rotem, and her fellow research partners. So she said it really does a joint effort, and she enjoys working with the whole group. She also reflects and says that, of course, every scientific endeavor has its ups and downs. So she makes a habit of celebrating the small moments of success and not only the finish line. So thank you, Nitzan, for sharing that wisdom and your really exciting science with us.
It's great. I'm sure she's enjoying her time with her child, but I bet she can't wait to get back to phages.
Well, she'll be able to, while she's taking care of the little one, she can listen to this episode of TWiM and feel really great about the highlight you gave her, Vincent.
All right. Well, thank you for that great work. We look forward to more, and that is TWiM number 270. You can find the show notes at microbe.tv/TWiM. Send your questions and comments to twim@microbe.tv. And if you enjoy our work, consider supporting us financially. You can go to microbe.tv/contribute and see the different options we have for contributing. Michelle Swanson's at the University of Michigan. Thanks, Michelle.
Thank you. My pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.